So, good morning, students. Today we will discuss another topic. It's interesting, a quite interesting topic. Today we are discussing. So, what's the topic's name? Glycolysis. Glycolysis. So, simply understand the term of glycolysis. Glycolysis. Glyco means glucose. Lysis means breakdown. Breakdown of glucose is termed as glycolysis. Is it incomplete or complete? It's partial partial breakdown. It's a partial breakdown of glucose C6H12O6 into two molecules of pyruvic acid. So if it was a complete breakdown, then we had got two products, or majorly three, that is carbon dioxide, water, and energy. But we are getting two molecules of pyruvic acid. From this, we can see that it is partial breakdown. Very important step to be analyzed or point. And in NCRT, it is mentioned that it is also called EMP pathway. So you will ask me, why this term EMP? E stands for a scientist's name. M Emden and Mar. What's Mio Hoff and N Parnas, I guess. Parnas. So from this EMP pathway, the name is given to this process, glycolysis. So now let's see the chemical reactions and the reactions which are taking place in glycolysis. But before that, this is very important, that this is an universal pathway. Means all organisms follow this pathway. It is a universal pathway. Star points I have mentioned here. Direct question can be framed. Next, let us see the reactions accounted in this process. So let's consider in case of a plant cell. You know that we have studied photosynthesis in the earlier unit, photosynthesis in higher plants, that plants produce or store starch or sucrose. So now let's convert sucrose, a 12 molecule carbon compound, into glucose and fructose. How you can convert sucrose into glucose plus fructose with the help of an enzyme, invertase. Very important step. First we took sucrose and converted into glucose and fructose by invertase. First step is done. And in NCRT it is mentioned that both glucose and fructose both can undergo glycolysis, but we have to study only for glucose. The first step is cleared, simply cleared. I'm rubbing it. So now let's start with glucose. The board is quite short, so I am dividing it into two. That is 5 steps here and 5 steps on the next page. That is on the next side of the page. So you will ask me, glycolysis is a how many step process? It's a 10-step process. Means first initial steps are called activation of C3H12O6, activation of glucose steps, and second step is called payoff step. That we will say how it is payoff. So glucose is getting converted into what? Glucose 6 phosphate. Glucose 6 phosphate. How it is getting converted? It is utilizing ATP and getting converted into ADP, because you can see a phosphate addition here, a phosphate addition, thereby ATP is converted into ADP. Enzyme here, hexokinase. It's an important enzyme highlighted in your NCRT. Done. First step is simple. Now in second step, very simple step. Glucose 6 phosphate is simply converted into fructose 6 phosphate. Simply fructose 6 phosphate. So you know that glucose and fructose have same chemical formula. You are just changing the structure. Means we are using isomerism formula, which is isomerase enzyme. Isomerase like enzyme you are using. Next, see, important. From this, we are converting into fructose. Very important. Fructose 1,6 bisphosphate. 1,6 bisphosphate. So again you got here a term bisphosphate. First it was only a single phosphate. Now bisphosphate means one more phosphate is added. How phosphate comes? By breakdown of ATP. So let's break ATP into ADP. ATP is broken down into ADP. Simple. Now here in NCRT it doesn't mentions enzyme, but in state board book it mentions here an enzyme. What is that? Let's see. In NEET JEE it is also required, so I am writing phosphofructokinase. Simply you can remember phosphate group is there, fructose is there, and simply it's the formation. By that you are remembering it. Not a challenge here. Now, so this is the activation. What is this? Activation of glucose. This major step, activation of glucose. We are activating the glucose. Now this is the 6 carbon compound. Till now all of these are 6 carbon compound. Now we are breaking them into two parts. Breaking them into two same parts. What I will say? Triose phosphate. From the term triose we have studied in biomolecules. Triose, tetrose means triose 3, tetrose 4, pentose 5, hexose 6, pseudoheptalose 7. In SSC they have mentioned that. But triose simple 3 carbon. And here also triose phosphate. But in the first triose phosphate, we are getting 3-PGL. 3-PGL. What is the full form? 3-phosphoglyceraldehyde. 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 And here we are getting DH AP. What is the full form? Note down. I am not writing because there is no space here. Dehydrogenase. Dehydroxy acetone phosphate. Dehydroxy acetone phosphate. So now note till this. Note till this. Wait. Stop. Pause for a minute. One more important thing, that this is not going to independently follow the process. This is getting converted into PGAL. Now PGAL will follow the individual process. Means after this, the products into two we are going to write. Two products will be formed because here they are separated into triose phosphate, two triose phosphate. Now this is getting converted into this. So subsequently it is understood that two products of next reaction will be formed. Now note down this. So pause and note. Okay. Now, what was our product? Yes, 3PGAL. 3PGAL, because DHAP converted into this. So this is our major product here. Now, what will happen? This is a very important step. Star to it. Now, and H3PO3, H3PO4 will give its phosphate. It is not an enzyme. It is just giving its phosphate group somehow. By giving phosphate group, energy is produced, and by this energy, very important, by this energy, NAD plus is getting converted into. It's getting converted into NADH plus one proton, H plus, and the product form will be, guess the product, two molecules. I told you, as trios, two trios were formed. This will also, the products will also into two will be formed. So 2 1 3 1 3 1 3 B PGA. BPGA means bis. 1 3 bis phosphoglycerate or bis phosphoglyceric acid. Simple. This was simple. Now next, this will just convert ADP into ATP. Means now it wants to get rid of the phosphate group. So it will convert ADP into ATP. So what will be the product? 3PGA. Simple, 3PGA. In competitive exam, enzyme is given here. What is that? Simple to understand. Phosphoglycerate kinase. By the name it suggests phosphoglycerate is formed, so the enzyme name is also like that. Then again, see, just it is doing isomerism, just like that. From glucose 1,6 phosphate, from glucose 6 phosphate to fructose 6 phosphate. Just like this is also doing isomerism. Just from 3PGA it's becoming 2PGA by using an enzyme which is called phosphoglycerate mutase. Just mutase means just there is a shift in the number. After that, 2 pga 2 pga will remove water. By removal of water, it is going to form PEP, that is phospho that is phosphoenol phosphoenol pyruvate. So this step is done. Now in this step, PEP will lose its phosphate, that is ADP is converted into ATP. It wants to lose its phosphate group because phosphoenol pyruvate, it wants to lose its phosphate group. So it will form pyruvate using enzyme pyruvate kinase. Simple name for the enzyme. So here also two molecules, it's understood, and 2 molecules of pyruvate. So this is the last step. The formation of pyruvate is the last step of glycolysis. Very important. So this whole is called payoff phase. Why? Because the utilized ATP are synthesized again. That's why it is called payoff phase. Note down this, then we will study the energy account. So okay. So what is the energy account? So in first that step, that activation of glucose, we saw that 2 ATP were lost, minus 2 ATP. Then in the payoff phase, what happened? 2 NADP were formed and 4 ATP were formed. So what is the net? Tell me the net. Yes, right. 2 ATP and 2 NADH. And we will see in Krebs cycle that how 2 NADH will form 3 into 2, that is 6 ATP. Means 1 NADH forms 3 ATP. We will see that in Krebs cycle, not in today's class. So this was glycolysis, a very important topic. Now in your NCERT, a small topic fermentation is also there. Let's cover this fermentation here in this class itself. Fermentation. So when fermentation takes place? After glycolysis, when less oxygen is available. Fermentation takes place. There are two types of fermentations: alcoholic fermentation or lactic fermentation. Lactic acid fermentation or lactic fermentation. In short, we can say alcoholic fermentation, pyruvate is converted into, it's 3 carbon, it's converted into C2, sorry, CHCHO means the aldehyde, acetaldehyde, acetaldehyde plus carbon dioxide means there is removal of carbon dioxide here in the first stage. This is about alcoholic fermentation, very important. Alcoholic fermentation I am talking about, alcoholic fermentation. So what will be the enzyme used here? Very important question. The enzyme used here is pyruvate decarboxylase. Decarboxylase. Decarboxylase, very important. Decarboxylase. We want ethanol as the byproduct. So we have to deal this in two steps. First step was this, second step is this. So how we can get an acid? By doing oxidation. Let's remove a carbon hydrogen from it. We'll do a CHCO, alcoholic enzyme, alcohol dehydrogenase. We will remove hydrogen, so we will get C2H5OH. Achha, this formula is wrong, sorry for the formula. It's acetaldehyde. Here also acetaldehyde. Yes, ethanol is formed. Ethanoic acid, that is, sorry, ethanol is formed from acetaldehyde. Ethanol is formed. We don't want acid here, we want ethanol as the product. So ethanol we got. This takes place generally in yeast. Let's see lactic acid fermentation, because in this topic there is not much important portion. Lactic acid fermentation is very easy. Lactic acid fermentation, so in which pyruvate is getting converted into lactate or lactic acid simply by using lactate dehydrogenase. One step process directly. This occurs in muscles of humans. Lactic acid is the byproduct, I can say. Lactic acid is the byproduct, and very less amount of ATP is produced, but ATP is produced in faster speed, so that the energy needs could be met. But very harmful or toxic products are formed, that is ethanol in case of alcoholic fermentation and lactic acid in case of lactic acid fermentation. So in this class we have covered two topics, glycolysis and fermentation, of which glycolysis is more important. Fermentation, just read the NCERT. If you have any doubts, just you can ask me on the comment section. So this was for today's lecture. Thank you for watching.